Today we're going to talk about blood, the parts of the blood, and the functions of the blood. So let's start with the functions. Of course, it's there to transport nutrients and oxygen, to get oxygen to your cells, to get carbon dioxide and other waste materials away from your cells. Also to transport vitamins and minerals and other nutrients from the digestive system to the cells of the body. It also includes immune cells and helps transport those immune cells to the site of an infection or damage and helps to fight infection. And then it has a really big role in regulating your body temperature and the pH levels of tissues, but the pH levels of the blood are also very important. We're not going to talk about that so much today. Just keep that in mind. That's something else that the blood does. When we take a sample of blood, you can then spin that in a centrifuge, which will separate it by density. And the blood separates into 3 layers. The least dense layer is called the plasma. And the plasma is about 55% of the volume of the whole blood. It's mostly water. And dissolved in that water are proteins and other solutes such as electrolytes, those are salts, other nutrients, minerals, vitamins, fats, amino acids, also the respiratory gases, oxygen, carbon dioxide, but also nitrogen, which we can't use, but that's what most of the air we breathe is, and then waste products being removed from tissue. So all of that is in the blood plasma. Also in the blood plasma are antibodies, which are signaling proteins used by our immune system. We're going to come back to those in another chapter, but just know that that's part of the proteins in the blood plasma. The next layer is called the Buffy Coat. Get it? It's your defender, like Buffy. Okay. The Buffy Coat, it's a very small portion of the volume of the blood, but it's very, very important. It includes the white blood cells or the leukocytes. There are 5 types of white blood cells. All these are immune cells which respond to any kind of pathogen or damage. And these that have little granules in them, those send out signaling molecules to attract other white blood cells to the site. Neutrophils are macrophages that engulf pathogens and destroy them, and then others are going to release antibodies to bind to any kind of pathogen. And then the other thing in the Buffy Coat are platelets. There are gazillions of platelets in every cc of blood, and platelets are actually just little portions of cells. We're going to come back to them. They're going to be really effective and important in damage to blood vessels. These are the 5 types of white blood cell. They're all derived, from bone marrow, one type of germ cell in the bone marrow. And you don't need to identify the different types of white blood cells, but you do need to know that they're all involved in fighting infection and repairing damaged tissue. Now, platelets and the white blood cells are all made by these cells in the bone marrow that do mitosis and produce new cells that become the white blood cells, the red blood cells, and the platelets. Platelets are formed by these megakaryocytes that create more of their own plasma membrane and cytoplasm. And that cytoplasm has special proteins in it. And then as these pieces of cytoplasm ooze out through pores in the blood vessels, they get pinched off and they become the platelets. So platelets are not cells, they're little parts of cells. When a blood vessel is injured, the platelets actually stick to the injured blood vessel. The platelets cannot stick to the cells that line the blood vessels. What they stick to is the collagen in the basal membrane that those epithelial cells stick to. So remember epithelial cells, they're always attached to a basement membrane. These are simple squamous epithelial cells called endothelial cells because they're the innermost layer of a blood vessel. And these are obviously cellular. Their surrounding is a plasma membrane, but the basement membrane they're sitting on has a lot of collagen in it. Platelets stick to collagen. So when the wall of a blood vessel is damaged, the platelets automatically stick to that collagen and then they become activated. That begins the clotting process. When platelets become activated, they start releasing this protein called fibrin that creates long fibers and traps the platelets and red blood cells. And that creates a clot or a scab. So this is an electron micrograph at 5,000 times magnification. So you can see all those structures. So here's another awkward Yeti cartoon with a couple of little platelets stuck together saying, "Platelet party!" You can see the little red blood cells in here as well. Then another one comes along and says, "What's going on here?" And they say, "Platelet party!" Now there's a bunch more stuck. And then there's more and more stuck with some red blood cells stuck in here. You can see, "Why, I haven't had this much fun in years. Platelet party! I think we're stuck. Platelet party! No, seriously, we're stuck. Platelet party!" Because being stuck together is a platelet party. And that's how you get a clot or a scab. Okay, so that was all the Buffy coat. Platelets and the white blood cells, or leukocytes. And then the red blood cells make up the densest part of the blood. They're about 44% of your whole blood. And they're called erythrocytes. Erythro means red. So erythrocytes are the red blood cells. And there are 4 to 6 million in every cc of blood. So you have 5 liters of blood altogether, about. So there are so many red blood cells in your body. Erythrocytes, like the other blood cells, are formed in the red bone marrow. And then they circulate through the bloodstream only for about 120 days. And the reason is that they don't have their own nucleus. They've actually pinched off the nucleus after they're made. The liver, and especially the spleen, are constantly monitoring the red blood cells. And old or damaged erythrocytes get phagocytized within the liver and the spleen. And then the parts of those red blood cells get recycled into the blood. The heme is a protein that is at the center of the hemoglobin molecule that is within the red blood cell. That's going to be converted by the liver into bilirubin and secreted as bile into the digestive system. And we'll come back to bile when we do the digestive system. Iron, the ions that were at the center of the heme molecule, those are going to be stored into the liver and then released back into the bloodstream to get sent back to the bone marrow to make more red blood cells. But your liver is constantly storing lots and lots of iron, which is why the liver is a very healthy organ to eat, because it does have lots and lots of iron and other minerals stored up in it. All the other parts of the cell, all the proteins, amino acids, lipids, all of those are going to go back into the bloodstream, and a lot of those are going to go back to the bone marrow to make new red blood cells, and the cycle starts over. So here is a poem from a children's book called The Blood Hungry Spleen. So here's the little red blood cells, and there's the spleen. And this is an odd poem, but hopefully it'll help you remember the job of the spleen. To the left of your stomach, a deep violet red filters at work, filling blood cells with dread. The red blood cell graveyard. It's not Halloween. I'm talking about that blood basher, the spleen. It's the blood-hungry spleen. Blood cells better beware. Just enter in here and you exit out there. But not if you're sickly. What's that I hear? It's the sound of sick blood cells all screaming in fear. It's 5 inches long and it's 3 inches wide. If you're an old blood cell, you best run and hide. And if you're a tired blood cell, you best stay in bed. It's the blood-hungry spleen, and it wants to be fed. So that's just a really silly way of reminding you that the spleen's job is to recycle red blood cells and to break down old or damaged or infected blood cells. The spleen is also a really important part of your immune system. So this is another awkward Yeti cartoon. about spleen. I am not beloved. I don't need to be. I filter old blood. I fight bacteria. I am a silent protector. I am spleen. So just FYI, you can get Spleen and the other characters from the Awkward Yeti cartoons on merchandise at the Awkward store. And no, I am not paid by the Awkward Yeti. I just love the cartoons so very much. All right, so when we look at a smear of blood, we can see the erythrocytes, which look pink, and they actually look hollow. And that's because they have no nucleus. They appear. It's supposed to say they appear hollow. The leukocytes are distinguished from each other by the shape of the nucleus. And then the platelets are these little tiny things. They're so tiny because, remember, they're not full cells. They're just little parts of cells. So the platelets are those little tiny things. You can see all of that in a blood smear on a microscope slide. Red blood cells themselves are what we call biconcave, meaning that the top and the bottom are both concave. They are round cells, and their shape enables them to bend while they're going through the capillaries. I will fix that right now. There we go. Okay. So the capillaries are the smallest of the blood vessels, and that's where gas exchange is actually going to happen, where oxygen leaves the bloodstream and carbon dioxide enters the bloodstream. So because red blood cells have so much surface area, and because they can actually bend as they go through the capillaries, a lot of that surface area can touch the walls of the capillaries, and then the gases have to have a shorter distance to diffuse. Some diseases cause red blood cells to clump up together in what we call a rouleau. So this is a bunch of red blood cells that instead of passing through individually like this, like they're supposed to, they've clumped up together. And obviously that's really bad for your circulation. This is going to cause pain and a lack of blood flow, so it can cause cramping in muscles. Your skin can turn blue because you don't have blood flow, and then it can cause death if it isn't eased. Sickle cell disease causes red blood cells to form this sickle shape instead of this nice round shape. In this sample, you can see that the blood cells are actually stuck together. And that's because this is a student's blood, a former student's blood, and we just took her blood and stuck it to a microscope slide. So it was actually kind of drying out by the time we looked at it. So that's why they're all stuck together like that. But you can see this cell right here in this little sickle shape. And as you can imagine, those also don't pass well through capillaries. And so, again, it can cause a lack of blood flow to tissues, and it can lead to death. All right, so within each of the red blood cells, there is a molecule called hemoglobin. Hemoglobin, there's a 4-part molecule. It is a giant protein made out of 4 proteins. And within the center of each of those is a heme, which is this thing here, which is a molecule with a space at the center for an iron ion. That iron ion can bind to an oxygen molecule. So the heme group binds to oxygen. It binds to carbon dioxide less well. So most carbon dioxide is actually just dissolved in the blood plasma. But each heme can bind to 1 oxygen atom. So each hemoglobin molecule can carry 4 oxygen atoms. Okay. In sickle cell disease, the difference between regular red blood cells and sickle cell is a difference in 1 amino acid in one of these protein chains. It's a really tiny change, but the shape of the hemoglobin molecule changes. That can change the shape of the red blood cell. All right, let's talk about one other thing in the red blood cells, and that is the antigens on the surface of the red blood cell and the antibodies in the blood plasma. And these give us our blood types. First, we're going to talk about the A, B, and O blood types, and then we'll talk about the positive and negative. That's a separate blood typing system. So Let's talk for a moment about your immune system. An antigen is anything that will cause a response in your immune system. So anti meaning against and gen meaning create. So this creates some kind of anti-response. So if you're type A blood, your erythrocytes have this certain molecule on the surface called antigen A. Blood type B has a different shaped antigen. Blood type AB has both, A and B. Blood type O has neither. Now, in the blood plasma of type A blood has an antibody that binds to the type B antigen. Now, antibodies are these special proteins that are really important in your immune system that have a stem here that can bind to a white blood cell, and then two branches specifically shaped to bind to a specific antigen. Each type of antibody binds to a specific antigen. So when we talk about antibody testing, we're testing to see if somebody has the antibody that specifically binds to a certain, for example, pathogen, such as a bacteria or a virus. And because these antibody binding sites are very, very specific, we can see if someone has been exposed, for example, to a virus by seeing if their immune system is making the antibodies that bind to that virus. Okay, and this is something your immune system makes after it's been exposed to something. All right, so back to the blood types. So if you take type A blood and you put it into someone with type B blood, then the anti-B antibodies bind to these molecules on the B blood cells. And notice there are two binding sites on each antigen, so this can bind to two different blood cells, and those blood cells start to clump together. Same thing, the antibodies in the plasma of the type B will bind to the molecules on the A blood cell, and again, they will clump together. So this is what happens. If we have the donor blood type of A, and the recipient blood type is A, then everything is fine and dandy. The antibodies don't have anything to bind to, and the blood is fine. If, however, you take donor blood type A and you put it in a recipient whose blood type is B, then their antibodies in the blood plasma start to bind to the red blood cells, and the red blood cells start to clump together or agglutinate. And as you can imagine, agglutinated blood does not flow well through blood vessels, and the person will die unless there is some treatment for it. But generally speaking, this is going to cause death. So now, looking at a B blood, they have no antibodies in their blood plasma. Type O, on the other hand, has antibodies for both A and B antigens. All right, so here's the thing. If you go give blood, and if you are able to give blood, you absolutely definitely should give blood. It is one way to save lives, really without making much of a sacrifice on your part. If you can give blood, if you have type O blood, then they say that you are the universal blood donor. But if you look at this chart for just a moment, you can see that that's not actually true. Your type O blood is not a universal donor for whole blood. It's a universal donor for red blood cells. And the reason that that's really important is that it's very hard to make red blood cells in a lab. We can make things that are sort of like blood in a lab, but red blood cells are you just need to get them from people. And so when you give blood, what they do is they take that blood and they spin it in a centrifuge and divide the 3 layers so that type O red blood cells can be given to anyone because they have no antigens. So it doesn't matter what the recipient's blood type is. There's nothing for antibodies to bind to. So you can give type O red blood cells to any recipient. So O is the universal red blood cell donor. But if you look again, you'll see that type O blood plasma is not a good plasma. You can only give it to people with type O blood because it's going to bind to every other blood type. So what then is the universal blood plasma donor? It's AB. Since they don't have antibodies, their blood plasma can be given to anyone. So O is the universal red blood cell donor. AB is the universal plasma donor. Plasma, you can actually give, I think, every 3 days or so because it's mostly water. And your body restores, obviously, the water as soon as you get rehydrated. But it can make the proteins in the blood plasma very quickly. So people who are AB, they can actually donate blood a couple times a week. So that is something that is good to know. And if you are type AB, please, if you are able to donate plasma, please do that. All right, now the positive negative is, like I said, a separate type of blood typing, and it involves a different antigen. The Rh positive blood has an antigen on the red blood cells and no antibodies in the plasma. If you're Rh negative, you have no antigens and you have no antibodies unless you've been exposed to positive blood. That could happen, I guess, during a transfusion, although they do test for that. More commonly, that happens during pregnancy because the blood plasma of the mother travels through the placenta and exchanges materials with the plasma of the fetus. So remember that the fetus gets all of its nutrients and all of its oxygen from the mother. So some of the antibodies in mom's blood can travel into the fetus's body, and some of the antibodies from the fetus's body can travel into mom's blood. Mom's blood, if mom is negative and she has a fetus that is positive, then she could, in the first pregnancy, it actually doesn't develop a problem. It's the second pregnancy. Then she could have antibodies that could bind to the fetus's blood. And that can actually cause problems. And so we now have a drug that we can give to pregnant mothers who have negative type blood to prevent them from developing those antibodies. Okay, so that's what those blood typings mean. So give blood if you can. And remember, erythrocytes have antigens. Plasma, remember, is mostly water, and that's where your antibodies and blood gases travel. And that's the end of our blood chapter.